Welcome to part 1 of the plant diversity video, Fungi presented by Bala, on behalf of Achyut or BAM-Sir. This video focuses on the fascinating world of fungi. The term fungus is derived from the Latin word for mushroom. The study of fungi is known as mycology. Pierre Mickley is considered the father of mycology, while Anton de Berry, also known as the father of plant pathology, is the father of modern mycology. Fungi are heterotrophic thallophytes and are eukaryotic and chlorophyllous decomposers. They are non-vascular and spore-bearing. Their reserved food material is glycogen and oil drops, and their cell wall is made of chitin, except for oomycetes, which have cellulose. The plant body of a fungus is called the mycelium. Mycelium can be classified astulose, filamentous, or branched. The mycelium is composed of a network of thin threads called hyphae, which collectively form the vegetative part of the fungus, leading to the fruit body. Mycelium is the vegetative part of a fungus, made up of a network of thin threads called hyphae. The unit structure of mycelium is hyphae. Mycelium is divided into two types based on the presence or absence of septa: septate mycelium found in higher fungi like Fusarium, and choanocytic mycelium in lower fungi like Albugo. Septa contain pores, which can be simple, a small opening in the hypha, or doliopore, which is a septum with a bulge in the center. Here we can see diagrams illustrating the simple pore and the more complex dolipore septum structure in fungi. Fungi exhibit different tissue-like structures, such as prosenchyma, pseudoparenchyma, sclerotium, and rhizomorphs. Similarities between algae and fungi include a thallose plant body, meaning no differentiation into root, stem, and leaves, and the absence of vascular tissues. Both have unicellular and non-jacketed sex organs. Embryo formation is absent. Asexual reproduction is by mitospores, and the nature of their nucleus is eukaryotic. Key differences exist in their habitat, chlorophyll presence, mode of nutrition, reserved food material, cell wall composition, type of parenchyma, and sex organs. Fungi habitats vary greatly. Coprophilous fungi grow on dung, epixilic fungi on wood, keratinophilous fungi on hair, nails, and hooves. Monoblepharis and saprolegnia are found in aquatic conditions. Fungi nutrition can be saprophytic, such as mucor, rhizopus, and penicillium. Parasitic forms include Puccinia, an obligate parasite, and rhizopus, a facultative parasite. Symbiotic relationships are seen in lichen and mycorrhizal roots. Reproduction in fungi occurs through three main methods: vegetative method, asexual method, and sexual method. Vegetative reproduction methods include fragmentation, where an organism breaks into parts capable of forming new individuals, cell division or binary fission, budding, and oidea formation, the production of asexual spores under stress. Reproduction in yeast by budding involves the formation of a developing bud, which grows into a new bud, eventually forming a chain of buds. Asexual reproduction methods utilize various spores. Zoospores are flagellated and motile. Conidia are asexual spores not enclosed in a sac. Aplanospores are non-motile, asexual spores. Sexual spores include ascospores and basidiospores. This diagram illustrates the various types of fungal spores, including zoospores, sporangiospores, chlamydospores, oidea, conidia, ascospores, basidiospores, acidiospores, uretospores, and teludospores. Sexual reproduction in fungi includes planogametic copulation, the fusion of two motile gametes, gametangial contact, the physical touching of gametangia for gamete transfer, and gametangial copulation, the fusion of gametangia. Planogametic copulation is the process of fusion of two motile gametes during sexual reproduction. Gametangial contact is characterized by the physical touching of gametangia for the transfer of gametes. Gametangial copulation is the process where the gametangia themselves fuse for sexual reproduction. Spermatization involves the transfer of male gametes, or spermatia, to female structures for fertilization. Somatogamy is the fusion of two non-reproductive somatic cells in sexual reproduction. Somatogamy is the fusion of two somatic cells, which are non-reproductive cells, during sexual reproduction. The kingdom Mycota is classified into Myxomycota and Eumycota. Eumycota is further divided into Fungi imperfecti and Fungi perfecti. Fungi perfecti includes Phycomycetes, Ascomycetes, and Basidiomycetes. This image provides a visual overview of the different classes and subclasses within the kingdom Mycota. Myxomycetes are a connecting link between protozoa and fungi. They lack a cell wall, have a naked amoeboid plant body, and are excellent for studying protoplasm. Phycomycetes, also called algal fungi or black molds, are considered primitive fungi with choanocytic hyphae, meaning they are aseptate and multinucleate. They are divided into oomycetes and zygomycetes. This concludes the discussion on Myxomycetes and Phycomycetes, introducing the first two classes of fungi. The third class of fungi is Ascomycetes, also known as sac fungi. Their hyphae are septate and multinucleate. Asexual reproduction occurs through exogenously produced conidia, while sexual reproduction is by gametangial contact, leading to the formation of the ascus. The ascocarp is the fruiting body of Ascomycetes. Examples include Penicillium, Aspergillus, Neurospora, and yeast. Ascocarps can be cup-shaped, flask-shaped, or closed-shaped. Next up is the fourth class, Basidiomycetes, or club fungi, considered the most advanced fungi. Their hyphae are septate and uninucleate, featuring doliopore septum and clamp connections. Sexual spores are produced exogenously in the basidiocarp, and sex organs are absent. Examples include Puccinia, Ustalago, and Agaricus. This image highlights some characteristic features of Basidiomycetes. The fifth class is Deuteromycetes, also known as fungi imperfecti. They are called this because the sexual stage is absent. Asexual reproduction occurs through conidia. Examples are Alternaria and Fusarium. This image illustrates the various classifications of the kingdom Mycota. We will now examine the life cycle of mucor, a type of fungus. Mucor, known as black mold or pin mold, is a saprophytic fungus. It features doliopore septum and a coenocytic mycelium, which means it is multinucleate without septa. This image provides a closer look at the mucor fungus. Reproduction in mucor is next, highlighting the different methods used by this fungus. This slide displays the asexual and sexual reproduction processes in mucor. We now see a comprehensive diagram of the life cycle of mucor. This image further emphasizes the detailed stages of the mucor life cycle. Here is a cross-section of the mucor fungus, showcasing its internal structures. Finally, we examine the complete life cycle of a mushroom, a common example of Basidiomycetes.